Our next speaker is Dr. Yan Sung. Dr. Sung is an associate professor at the Emory University Rollins School of Public Health. Dr. Sung completed his PhD in biochemistry and molecular biology, as well as a master's in computer science at Wayne State University. Prior to joining the faculty at Emory in 2011, Dr. Sung held the position of assistant research professor at the School of Public Health at the University of Michigan. His research interests focus on novel study designs and applications of genetic epidemiology to identify molecular markers and understand risk factors and functional mechanisms of human diseases in multi-ethnic populations. The goal of such translational research is to improve the prediction, prevention, and treatment of human diseases. Dr. Song is the director of the Genetic and Molecular Epidemiology Certificate Program at Rollins School of Public Health. Please welcome Dr. Song.
Hello. First, I want to thank the organizers for inviting me and giving me the opportunity to share some thoughts and experience about multi-omics research in population studies. I have no conflict of interest to disclose. So this is an overview about the role of genetics and molecular biology in human health. If you remember the central dogma, the genes coded in DNA can be expressed under epigenetic and transcriptional regulation. Then the messenger RNAs translate into peptide and the proteins. The small molecule metabolites interact with these macromolecules in pathways and networks to play important functional roles. Such genetic and molecular functions and their variation at the cellular, tissue, and organ levels can represent inherited effects as well as response to the changing environment. In recent years, this research of microbiome helps us understand the functional roles of these coexisting microbes on overall human health as well. The technological advancement allows us to measure all these molecular layers using high-throughput approach. We have developed methods and approaches to investigate the genome, epigenome, transcriptome, proteome, metabolome individually in relation to many human diseases. Now it's possible to investigate the biological systems jointly that can capture the short-term and long-term response to the environment. The inter-individual variation in molecular systems across populations may hold the key for a holistic understanding of host genetics, pathogens, and the environment in health and disease at the population level. Most of my current projects are rooted in this framework of how genetic and molecular factors contribute to health and disease in populations, and how genes and molecular pathways interplay with environmental factors. We're all sort of biased. However, I want to make a case that the discovery research or hypothesis generating research can be very effective when we have little or incomplete knowledge. Discovering the interesting relationship between molecular markers and the disease traits is critical first step, which can lead to more focused hypothesis testing research to ultimately demonstrate molecular functions. And we can investigate individual molecular layers effectively using established association analysis methods, but none of them can fully represent the biological and the population variation, since DNA, RNA, protein, and metabolites often have complementary roles to jointly perform a certain biological function. Such complementary effects and the synergistic interactions between molecular layers in the life course can only be captured by integrative studies of multiple relevant molecular layers. When we scale up such molecular layers relationship to the omic levels, we have the opportunity to understand the whole systems altogether. Genetic association study is a powerful tool. I'm a geneticist, and that approach can understand the heritable components of disease and traits. but it cannot explain time-varying risk factors for diseases. For most diseases, they have both genetic and environmental components. This figure from a review article nicely summarizes the complexity of environmental influence on disease development. In this example of arteriosclerosis, we can see that many risk factors and protective factors can influence the disease trajectory at a different time point and with different duration. Therefore, how to measure those environmental exposures and to capture their biological effects on disease development is a very important topic. So far, a large proportion of phenotypic variants driven by non-genetic factors remain unexplained. The multi-omic studies have the potential to transform the research of the environmental exposure and the biological responses to the environmental effects underlying disease development. The analytical methods for multi-omics research are not trivial. Several early review papers, including ours, discuss several options to explore different types of research questions. Machine learning and artificial intelligence methods are good at predicting and classifying the outcomes. They can be very efficient in variable selection using high-dimensional reduction algorithms. There are also methods for pathway enrichment analysis, and the network analysis can identify and summarize very complicated biological relationships based off all kinds of connections between molecules and disease traits. Today, I will only focus on a class of methods addressing potential causal relationship and the molecular mechanism, including Mendelian randomization. I will also briefly show you an aggregation approach as a potential omnibus test based on individual associations across multiple omics layers. One intuitive modeling approach to integrating genomic and other functional genomics data is to identify the genetic determinants of other omic markers, such as mRNA, proteins. These are so-called quantitative trait loci, QTLs. A QTL is a section of DNA that correlates with variation in phenotype, including molecular phenotypes, such as gene expression, metabolites, epigenetic markers. Omic QTLs can illustrate important molecular network mediating the interaction between the genetic variants and the environmental factors for human diseases. They can also be used as instrumental variables in Mendelian randomization studies of omic markers to illustrate potential causal roles. This figure summarizes billions of association tests between genome and DNA methylation, organized by chromosome positions of genetic variants and the DNA methylation markers. The red dots represent statistically significant pairwise associations after multiple testing correction. The right diagonal line indicates the so-called cis methylation QTLs, showing the associated genetic and epigenetic variations co-located in the same region. We also observe a lot of trans QTLs, which means the genetic loci regulating DNA methylation located far away, sometimes